In this video, we will be continuing our lecture on proteins. My sources in making this reviewer, uh, they're basically the Campbell 12th edition biology book and um, the corresponding video from Osmosis. Where we left off, we discussed about protein functions and basically everything else about proteins. The structure, basic definitions, general information, and yeah, many more. So yeah, let's recall. Proteins have their building blocks are basically amino acids, and um, they have diverse structures, which also is equivalent to having a wide range of functions. So protein functions consist of um, being enzymes, being responsible for the defense of um, being our defense against diseases, storing amino acids, transporting substances, particularly through or across the cell membrane, um, being components in hormones, particularly insulin, and being receptors, the low density lipoprotein receptor for bad cholesterol, um, particularly in the active transport, which is more specifically an example of active transport in regards to LDL, in the function of LDL receptor, is the receptor mediated endocytosis, if I'm not mistaken. And of course, we have the CD4 receptor and the CCR5 coreceptor. These are related to HIV, which I will be mentioning later. More on that later. And we have contractile motor proteins um, for movement, proteins that are seen in our muscles, and of course, structural proteins. They give support. Keratin for the hair, collagen for the bones. I'm not sure about elastin though, but yeah, as well as silk fibers. Now, there are 20 amino acids. So they are divided into three categorizations. First is the ones with nonpolar side chains, which are in turn hydrophobic. So take note that nonpolar substances are naturally hydrophobic. So these go along together. So when a molecule is nonpolar, then it's obviously hydrophobic. Hydrophobic, um, imagine oil. Oil does not mix well with water. It makes it then makes oil hydrophobic. So yeah, phobia basically to water. We have 1, 2, 3, 4, 5, 6, 7. We have 7 under this categorization of amino acids with nonpolar side chains. We have glycine, alanine, valine, leucine, isoleucine, methionine, and phenylalanine. So yeah, if you have to memorize the structures, then they're given here anyways. Go ahead and memorize them. And if you've observed, generally, the structure of amino acid, there is a general structure, which is basically this part. So every amino acid in this list contains that structure. And what is connected to, above depends on the particular amino acid. So for glycine, we have hydrogen at the top of this carbon. For alanine, we have CH3. For valine, we have, I'm not sure what this is called, but yeah. The bottom line is they have different structures at the top of the general structure. So they're all the same below, but what's above is different depending on what amino acid it is. Wait, we don't have 7, we have 9. Almost forgot to mention these two. We have tryptophan. I'm not sure if it's tryptophan, but let me double check. Tryptophan. I got the spelling wrong. I apologize for that. Tryptophan. Alright. So that's for tryptophan. And we have proline. So if you want to memorize, this is the order as per the Campbell Biology book. We have glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Now, for the second categorization, we have polar amino acids with polar side chains. Analogy. If nonpolar is to hydrophobic, then polar, which is the opposite of nonpolar, is to hydrophilic. That means polar molecules are naturally hydrophilic, which makes them, say, water-loving. They mix well with water. So, what we have under this categorization, we basically have 6. We have serine, threonine, cysteine, tyrosine, asparagine, or asparagine, whatever works, and glutamine. So again, there's a general structure, which is this. You can see this structure in every amino acid. Oops. And what is unique to the amino acid is the structure above. What makes the amino acid different from one another is what we have above. For cysteine, we have this. Threonine, we have this, etc. Okay, memorize for to memorize the amino acids with polar side chains, we have serine, threosine, cysteine, tyrosine, asparagine or asparagine, and glutamine. Now, for the third categorization, we have those with electrically charged chains, which are hydrophilic, just like the second category, the amino acids in the second category. So what we have here, we have we have 2 subcategorizations in this third category. We have those with negative charge, which are the acidic amino acids, and the ones with a positive charge, which are the basic amino acids. So for the negatively charged ones, we only have 2. They're basically aspartic acid and glutamic acid. And for those with a positive charge, we have lysine, arginine, and histidine. So that's it. This, by the way, is my reference. Basically, wrote it by my... I basically copied whatever is written in the Campbell ebook. This is page 77. Chapter 5, the structure and function of large biological molecules. So, yeah. If you can see the purple highlight, those are basically the general structures for each amino acid remains the same. What changes, what is different, is the structure above, one of the carbons, which is this in particular. The structure is unique to each amino acid. That's basically 20. Alright, let's move on. For polypeptides, they are the amino acid polymers. So the building blocks in the making of a protein is basically amino acid plus carboxyl group. They are all joined by dehydration reaction. This is basically the minus or a removal of a water molecule, and this would result in a covalent bond, which, as for proteins, the unique term is called peptide bond. And the repeated process involving cells, this process basically repeats again and again for the synthesis of polypeptides. Polypeptide is basically the polymer of many amino acids linked by peptide bonds. Let me recall what was mentioned in the previous videos on biological macromolecules. Biological molecules. Since lipid isn't exactly called a macromolecule, or considered even, since it's not a true polymer. Anyways, so for carbs, let's say polysaccharide. This is to glycosidic bond. For lipids or triglycerides, we have what is called the ester linkage. For polypeptide, we have the peptide bond. So that's all. Those are the three things you have to know. I might not mention the concept on nucleic acids. That's a different thing that it's related to this general lesson, but our teacher considered to not have that as our lesson anymore. But let's see, what is the bonding? What do you call the bonding of nucleic acids? Nucleoside linkages. Maybe that's the one. That's the term we're looking for. But anyways, I won't be discussing that much on nucleic acids. It's so far as I could go. So for polypeptide again, let's move on. The length is basically a few to 1,000 more or more. And each polypeptide has a unique linear sequence depending on what amino acid that is, depending on what protein that is. So the kind and sequence of side chains determines the molecule's chemical nature, which also ultimately determines what final shape the polypeptide is going to have. So in terms of the protein structure and function, specific activities of protein result from their intricate 3D structure. So as what I mentioned earlier in the opening of this lesson, the structure or shape of the protein will indicate what function it would exercise. So for instance, amino acid sequence of the polypeptide. That's one example of the 3D structure. The sequence will dictate what activity it will enact. So for the amino acid sequence of a polypeptide determines the protein's 3D structure. That's just related to what I said earlier. And the cell synthesizes a polypeptide, particularly the making of proteins in the cell. Something called the nucleolus is responsible for the synthesis of ribosomes, and those ribosomes eventually migrate to the rough endoplasmic reticulum, and in the endoplasmic reticulum, the ribosomes make the protein and those proteins eventually reach the Golgi apparatus for protein folding, and yeah, they get delivered eventually, I guess, in vesicles, if I'm not mistaken. Um, in the vesicles, the proteins are already supposed to have their own unique 3D structure and ultimately their sequence, which dictates their function. So a protein's specific structure determines how it works. So we have some proteins such as globular and fibrous. These are the types of proteins in terms of shape. For globular, it should look something like this. It looks like a globe in some degree. And for fibrous proteins, they somehow look like this, like long fibers. And these are just broad categories. There are many more countless variations since, as mentioned earlier, there are many structures and kinds of proteins. Even people who specialize in biochemistry couldn't memorize everything. Well, it's not really that important to memorize everything. But yeah, protein receptor molecules, we have something called endorphin. And for drugs, you have morphine, heroin, opiates. Proteins function as a receptor to bind another pain-relieving molecule. So this is somehow called the exquisite molecular order. So for the protein structural models, we have three. One is the space filling model. Two is the ribbon model. Number three is the wireframe model. So yeah, lysozyme is a protein that you find in tears and saliva. That's one thing you might want to know. So we have 4 levels of protein structure. 3 of them, primary, secondary, and tertiary, specifically, are superimposed levels. So for the primary structure, let's start with that. Primary structure is basically the linear chain of amino acids. Remember, always associate primary with linear chain. Linear. This is the sequence of amino acids. It is not determined by random linking of amino acids. It's not by random. It's determined by inherited genetic information. So yeah, there are fixed codes, genetic codes. So for the primary structure, it dictates the second and the tertiary structure. For instance, this is not an actual genetic code or sequence, but let's say 1, 2, 3, 1, 2, 3. So there's a specific pattern that is followed. It's not by random. It's not like 3, 2, 2, 3, 1, 1, 2, 3. You don't dictate the code. There is a fixed one. So for secondary structure, regions stabilized by the hydrogen bonds between atoms of the polypeptide backbone. And this involves interactions between backbone constituents. It coils and folds in polypeptide chains. And the result of H bonds between the repeating constituents of the polypeptide backbone, not the R group side chains. R group is basically whatever is going to be, say, if I recall correctly, the R group is basically what is unique about the amino acid. What structure is unique to that? I guess this is the R group, because we know that the ones below are general structures. Every amino acid has it. But what is unique to a particular amino acid is what's above, and this is called the R group. Kindly correct me if I'm wrong, but yeah, that's what I understand. So within the backbone, oxygen atoms have partial negative charge. Hydrogen atoms, on the other hand, attached to nitrogens have partial positive charge. So basically, one keyword for secondary structure is A helices and alpha helices and beta pleated sheets. Now, for tertiary structure, this is basically the one. Let me mention this to you straightforwardly. Tertiary, the keyword is 3D shape interactions between side chains. So as long as there's a mention of 3D shape, that's most likely tertiary structure. Again, for primary structure, linear chain, secondary structure, alpha helices and beta pleated sheets, tertiary, 3D shape, and interactions between side chains. So tertiary structure is basically the overall shape of a polypeptide due to the interactions between the side chains of the amino acids. So there's something called hydrophobic interaction. It indirectly contributes to the tertiary structure. You could also take note of this term. It's associated, it basically influences tertiary structure, hydrophobic interaction. This is caused by the exclusion of nonpolar substances by water molecules. These nonpolar substances are the amino acid side chains. So hydrophobic interaction is basically the London dispersion forces, or in other words, the van der Waals interactions. The hydrogen bonds between polar side chains and ionic bonds also stabilize tertiary structures. So if you hear about ionic bonding and hydrogen bonding, which is basically the same as H bonds is basically the same as van der Waals interactions. But yeah, those are all keywords for tertiary structure. Now, we have tackled the three superimposed levels of structure. Let's mention the last one, which is the quaternary structure. This is the association of two or more polypeptides. So two or more polypeptides. This is in some proteins only. The association of these polypeptides are supposed to form one functional macromolecule. So this is the overall protein structure resulting from the aggregation of these polypeptide subunits. I'm pretty sure aggregation is also a keyword for quaternary. Better watch out. So for instance, we have collagen. This is 40% of protein in the body. Collagen comprises of 40% of protein. Let's proofread that. Let's see if it's actually true. It's 30%, but it's mentioned it's 40% in the book. Maybe you say 30 to 40%. So we have transthyretin. This is for the union of four polypeptides. Also hemoglobin, the one that makes your blood color red because of a pigment called I'm not sure what it's called again for a while. Porphyrin. There. Porphyrin. That makes it's basically the red pigment in your blood, which is inside the hemoglobin. So basically, hemoglobin consists of four polypeptide subunits. You have alpha and beta. It's shared in alpha and beta. So that's it, I guess. Wait, I forgot to mention this about tertiary structure. We have disulfide bridges. They are covalent bonds. They further reinforce protein shape, and they form where two cysteine monomers are brought close by protein fold. I'm pretty sure this happens in the Golgi apparatus where protein folding occurs. And the cysteine, if you remember earlier, it is one of the um, proteins, one of the amino acids mentioned there, cysteine. Cysteine is one of the polar, um, one of the amino acids with polar side chains, in other words, hydrophilic. There. Alright. Um, More on the alpha helices and beta pleated sheets. Um, for alpha helices, they are delicate coil. They are formed by hydrogen bonding between every fourth amino acid. And for beta pleated sheets, there are two or more segments of polypeptide chain lying side by side, which are connected to hydrogen bonds between segments. They make up the core of many globular proteins. Again, globular proteins have the general shape of being like a globe, like that. And they dominate some fibrous proteins. So this is basically everything mentioned associated with tertiary structure. We have the hydrogen bond, we have the hydrophobic interactions, and the van der Waals interactions. I'm not sure why van der Waals and hydrogen bond are not the same. Please correct me if I'm wrong, but if I remember my chemistry from last year correctly, they're also the same thing. Maybe they're not. Let me double check. Hmm. Okay. They're different. Oh. Alright, they're different. I take back what I said earlier. Hydrogen bonding and uh, London dispersion are different. London dispersion, by the way, is the same as van der Waals. Now, uh, what do we do? Wonder what's next. So the change in primary structure by substitution of one amino acid, for instance, sickle cell disease. We all know that the primary structure is the linear chain of amino acids. There is a fixed code, right? There's a fixed code. And if you break that code, things could go wrong. One in particular, in hemoglobin, if the primary structure changes in hemoglobin, you could get what's called sickle cell disease. So the shape of red blood cells in your body are not normal. It's sickle shaped. So when you substitute, I think this is gly. I think it's supposed to be gly. That's the very first amino acid. There, glycine. Substituting glycine with valine, if I'm not mistaken, if I remember correctly. Submit gly with valine at the sixth amino acid position in primary structure of hemoglobin could cause sickle cell disease. So what gives? So what? It just proves that just basically proves how simple the change in protein structure can be devastating on protein function. So yeah. So what determines protein structure? For the most part, a polypeptide chain is arranged in a 3D shape depending on the interactions responsible for the secondary tertiary structure. This also depends on the physical and chemical conditions of the protein's environment. So, we have two terms before we end the video on proteins. We have denaturation. Of course, this is in the context of a protein. This is when factors get altered. Um, Weak protein bonds and interactions may be destroyed, causing protein to unravel and lose its native shape, or in other words, dissolve. Therefore, the denatured protein is biologically inactive. And denaturation is also when proteins are transferred from an aqueous environment to a nonpolar solvent. So, for instance, we have ether and chloroform. The factors, by the way, in a protein basically consist of pH, salt concentration, temperature, and other aspects in the environment. So, yeah. Denaturation agents, I'm still not finished with this, but these are everything that we have so far. Chemicals that disrupt the hydrogen bonds, ionic bonds, disulfide bridges that maintain the protein shape. If the disulfide bridge process is affected, what it produces, let's see, produces proteins. But when this is affected, their protein shape is not reinforced properly. So in that regard, um, an affected shape will also influence, um, will yield a defunct. Basically, the function wouldn't be as expected. It wouldn't be normal, which is going to be a bad thing. So, excessive heat. When the proteins are exposed in too much heat, that's why you have fever and lutong egg. Yeah, when you cook an egg, the protein composition in the egg changes. That's why it's no longer liquid inside, it's solid. So for renaturation, this is when the chemical and physical aspects of its environment are restored to normal. There are some processes of that cause denaturation which is irreversible, such as the one with the egg, I suppose. But in renaturation, there are some instances where you could still restore um, the proteins factors, the factors that influence the composition of proteins to normal. So let's see. Um, hmm. Hmm. So I think this supports my claim that eggs can no longer turn back to liquid um, once they are cooked or boiled, basically. So yeah. We're not done yet. We still have that puzzle. All right. We still got this. For proteins, for a while, let me just be sure if I'm late. Yeah, bro, I'm late. I'll continue this video. I guess let's call it a part 3.